Right, here we are with part 2 of our Agouti tale, and we've got some frightening looking information frankly on this page. We've got a mutant form of the gene now, so they're hoping that they're assuming you completely understand everything that's happened so far, and now we're going to introduce a mutant. And this was the A gene. Remember, we had big A, little a. And they've decided to call it A with a superscript VY, which results in hair which is yellow along its entire length. So remember, the agouti is yellow bands on the dark, on the black background, so that gives us bands. The recessive little a, well, we don't get any of these yellow bands due to the agouti gene being switched on. And then if the agouti gene is switched on continuously in this mutated version, then we get entirely yellow. So that kind of seems pretty straightforward, and we'd actually then go, okay, well, if we've got this mutant, then I'm going to have a yellow mouse. Pretty straightforward. Yeah, got the mutation. The gene is switched on continuously, that gives yellow, and we're going to have some yellow mice. End of story. No. We've got some mice here. I presume this is in colour on the original photo, but even in black and white we can see we've got a range of colour. We've got some very light coloured mice at one end, and dark coloured mice in the end. I don't know how they managed to get them lined up like that and stay still. They probably stuck them with glue. I don't know. The mind boggles. How did they persuade them to line up like that in order? Must have taken them hours.
Sellotape under the feet.
Sellotape them. These mice have identical DNA base sequences, but the gene is being expressed differently. This should set an alarm off inside your head going, oh, same DNA, but being expressed differently. I know how that might happen. Okay. And say, what name is given to the study of the control of gene expression by factors other than changes in the DNA sequence, and that is our epigenetics. So finally, we get a nice easy mark in this question. But even if you didn't understand the mice, just from that short stem of the question here, you'd have got epigenetics. So there is a slight moral to the story. Is that sometimes you can guess these, or from the definitions you can put these answers down even if you don't fully really get the diagrams that are given. And this next one is a case in point. This is all about, if you just look for things that you recognise on this diagram, you've got genes being switched on and off, and proteins being made, and RNA being made, that sort of thing. If you had to name processes that were to do with genes and RNA and proteins, there are a limited number of processes that you'd come up with. You'd probably, if you cover up that diagram and then guess, you'd probably say the two answers. However, here we go. We've got a gene that gives RNA. Well, what process is that? X is between the gene. Ignore that it's a regulator gene. Ignore that it's normal RNA. Gene gives RNA. That's got to be transcription, hasn't it? Transcription. And why RNA leading to the production of a protein? So RNA leading to the production of a protein, that's got to be the other one, translation. So I might not fully understand that diagram, but I can certainly get those two marks. What does this diagram show though? We've got our normal agouti situation at the top with the banded hair, with this gene that's going to give the yellow being switched on and off in a cyclical way. and with a mutation below we get abnormal RNA that blocks normal RNA, and so we don't get this regulator protein that makes it go on and off. So basically this just makes it stay on, because this regulator protein makes it go in a cycle, and we're blocking that. So we're just going to permanently get yellow hair. And so our last 3 marks over the page, it tells us what's going on, methylation, and it has been suggested that the methylation of the inserted sequence of bases could be responsible for the range of colours. Explain how. So that means we're going to sort of give our knowledge and try and make it relevant to this situation. How the addition of increasing number of methyl groups to the inserted sequence of bases results in lines and range of coat colours. The 3 marks. Okay. Let's give a general point about methylation. So methylation, sticking a methyl group on to DNA, makes DNA tighter, like more tightly wound around those histone proteins. So that's just from your knowledge, but it's kind of a AO3 situation where you are trying to suggest how things might work, but we're still going back to our knowledge. And so more methylation, and then explaining what might happen in this diagram. So more methylation, less blocking molecule, so less blocking molecule. And so you can look at this for the information, and you can go, if it's methylated you're going to get less produced. So mutation there, producing RNA, so that will happen more or less. So the more the gene is switched on and off, so more, that would lead to more methylation, less blocking molecule. I'll put in the more normal regulator protein, more normal regulator protein is made, and so the normal situation would happen more. So more the gene is switched on and off. So to kind of summarise that, more methylation, more agouti. So more methylation, more agouti. So on our diagram, methylation there would stop this process, and so our normal process of gene switching on and off cyclically would happen. And we finally got to the end. Well done.